right, well, good morning. So yesterday you guys looked at this schnapps and you made your character. So what we're going to start with today is to take a look at what everybody did. Take a look at each other's characters and do a comparison. You'll need to unsubmit your protein synthesis assignment so that you can finish those application questions. So you can just click unsubmit, do the application questions, and then resubmit it. And then together we are going to work on this transcription assignment in breakout rooms. And then you do have an optional knowledge probe that's linked on Google Classroom where you can read through kind of this process of transcription and looking at RNA so that you understand what that molecule is. So let's take a look at the schnapps. What you'll do is on Google Classroom, you'll look at your protein synthesis. And on that assignment, you have the slideshow where you added your character. So then what you're going to do is take a look at each other's to kind of see what's happening there. Now, as you're doing that, think about why they are the way that they are. So what determined if the organism was orange? What determined if the organism was blue? And why would one be different from another? So for example, like Mary Kate's is hairless, has 4 legs, no freckles, orange skin, short nose. Where TZ's has is hairless, non-freckled, 4-legged, orange skinned, and short nosed. Okay. This one is, you can't read the trace on that one. This one was hairless. Okay. And did not have freckles, has the 4 legs, a long nose. Some of them, if you go through, will have 5 legs, some are blue, some are orange. So why are they different? Okay, so thinking about where you got the information from. The other thing that I want you to make sure that you're recognizing from this is how did you get that information? So as we looked at it, you started with the DNA, right? So we start with the DNA. And then from your DNA, you determined RNA by saying that A bonded to U, G bonded to C, C bonded to G, T bonded to A, etc. And then you used that RNA to determine an amino acid. And we'll talk about amino acids a little bit more tomorrow. And then eventually you got your trait. Okay? And this trait is a protein. All right, so we're going to start to refer to traits as proteins. And we'll talk more about proteins as we kind of move on. This whole concept right here, though, is what we refer to as the central dogma of biology. So a dogma is kind of a set of rules, and it's like the meat of what it explains what's going on. And so religions all have dogmas to explain their religions and what their beliefs are. This is from the evidence that we have with biology. This is what we refer to as the central dogma because we know that all living things have DNA and that DNA structure is the same, and they all do this exact same process to get the traits that they are. And this then is what we can use to explain evolution, how organisms have things and do things, etc. So I want you to make sure that you understand that from this activity. Okay. The next thing that we then did in class was to take a look at this process of transcription. So really what we're focusing on is this first part of our central dogma, which is this right here. Okay, so we're focusing on this idea, which is transcription, and how we make DNA from RNA. Okay, so when we think about that or take a look at that with the activity, you'll open up a Jamboard. When you open your Jamboard, it looks like this. You'll use this first slide on the Jamboard to answer questions 1 through 7. Okay, and based on that, no, 1 through 6, sorry. So you'll answer 1 through 6 looking at those. Then I want you to watch this video, which explains the process of transcription, and answer these questions. Some of the key things that you'll find when you watch this is that you should see, oops, pay attention to you don't need to know all these transcription factors, but really it's just kind of watching this part of it. And what you'll notice is that it's only going to copy, this is the DNA. And so it's going to start here at this beginning. So it doesn't copy the whole piece of DNA. It only copies part of it. And as it moves on, then you've got, this is these transcription factors that get it started. You'll see this RNA polymerase, and that's bonding to the DNA here at the beginning. And then as it's transcribing, it's making the RNA. So the DNA is unzipping. This is the RNA, which is a complementary of just this side of the DNA. So it only copies 1/2, and it makes this piece of RNA, okay? So you'll understand what RNA is from those first 6 questions. And then, so you'll watch this, take some notes on it, and answer, that's what this box is for, okay? Then what I want you to do is go back to your Jamboard, and on the second slide, you're going to first build a DNA molecule that will look like something like this, where you put just some of the letters in together, pair them up with their complementary pairs. Okay, so I have, you know, C with G, G with C, C with G, A with T. So pair those up. So I'm starting with DNA. Okay, and what then is going to happen is I'm going to model how I could make RNA from this DNA. So the first thing that has to actually happen is that the DNA has to unzip, just like in our DNA replication. So these kind of come apart. They would stay together this way. It's not a perfect model, but so those unzip, and then we're going to just copy one strand. So this C would bond to an RNA G. And this G would bond to an RNA C. And this C would bond to an RNA G. Okay, so you're going to figure out your sequence there. Eventually then, if you looked at the video, what happens is that as this continues on, the RNA, the DNA goes right back together behind after it's been copied. See how this is all still one strand? So the DNA actually goes back together as the RNA comes off. So as this continues to grow, this is going to come off over here as my RNA, and my DNA would connect back together. So at the end of this, I still have my intact DNA, and then I have my separate piece of RNA that's a single strand. So this is the RNA, and then this is the DNA, okay? So you can take some notes on that, do your model, and then there's another video to watch, and this is actually, so you can watch this video. It does it in real time. See if you need to modify your model at all. Okay, and then you're looking at the structure here in terms of how they're the same or different. So that's the idea that the RNA is single-stranded, DNA is double-stranded, RNA has the U, DNA has T, but they still both have A, G, and C. Okay, and you're still bonding A to T or A to U, C to G, and G to C. All right. From there, then, this is on the 3rd page of the Jamboard. There's the 3 different types of RNA there. So describe their structure. After you've described the structure, you'll need to do a little bit of research about their function. So you can Google that, or you can look in the optional reading and get that information. And then answer these 4 questions here. What we'll do tomorrow in class is kind of pick up with this concept and then add on the idea of translation, which is the other piece. So hopefully that helps. Send me an email if you have questions, and I will see you the next time in class.